Welcome to The Explainer. Today, we're diving into a bacterium that's a real head-scratcher for microbiologists, Ligilactobacillus salivarius. It's a bug that has a split personality, and figuring it out is more important now than ever.
All right, so here's the plan. We'll start by looking at the two very different faces of this bacterium. Then we'll head into the lab to see how it gets identified. After that, we'll talk about when it turns from friend to foe, try to untangle the tricky treatment guidelines, and wrap up with the big challenges for infection control.
So this slide really gets at the heart of the problem. On one side, you have Ligilactobacillus salivarius being sold right off the shelf in health supplements, praised for promoting gut health. But on the other side, in the hospital, it's an opportunistic pathogen that can cause some really nasty infections. We're talking bacteremia, endocarditis, you name it. And the real kicker for us, it's naturally resistant to vancomycin. Now, first things first, let's clear up the name. You might know this bug as Lactobacillus salivarius. Back in 2020, though, the whole genus got a major shakeup based on genomic sequencing, and our bug became Ligilactobacillus salivarius. You're definitely going to see the old name floating around in papers or on your lab's automated system, so just be aware of that change. Okay, so what happens when this thing actually shows up on a culture plate in your lab? How do we nail down the ID? Let's walk through the whole workflow, from the classic gram stain all the way up to the latest genomic tools. So the workup starts out pretty standard. Under the microscope, you're going to see gram-positive rods, no spores, and they often like to hang out in chains. On the culture side, it's a facultative anaerobe. It really thrives in a CO2 incubator and grows nicely on something like MRS agar. Biochemically, the big one is that it's catalase negative, but, to get that definitive species-level identification, you've got to use the modern stuff. MALDI-TOF for sequencing is pretty much essential, because just relying on biochemicals can lead you astray. Now let's focus on the single biggest clue you're going to get. This isn't just some random resistance pattern. It is baked right into the organism's DNA. It's a fundamental part of what it is. And this is it. This is the aha moment for any microbiologist at the bench. If you've got a catalase-negative gram-positive rod from a sterile site and it's resistant to vancomycin, ding, 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 you should be thinking about the Lactobacillus family. And remember, this isn't acquired resistance. Vancomycin just plain doesn't work because the bug's cell wall has a D-ala-D-lactate end, not the D-ala-D-ala target the drug needs. It's a fantastic built-in diagnostic marker for us. So if this is a normal resident of our gut, what makes it go rogue? What pushes it over the edge from being a harmless commensal to a dangerous pathogen? Well, it turns out the answer usually isn't about the bug itself. It's about the host. Right. So this switch from good bug to bad bug is almost always about the patient's condition. In a healthy person, an infection is incredibly rare. But it's a whole different story for vulnerable patients. We're talking about people who are immunocompromised, who have just had major surgery, who have a central line, or especially premature babies. In these groups, this organism can be a serious, life-threatening problem. This case from 2018 is a perfect example of how this happens. You have a patient who undergoes gastric bypass surgery. He develops a leak where the gut was reconnected. Well, that leak is basically an open door, letting this gut commensal walk right into a sterile space. The result? A nasty abscess. And what's really interesting is that the lab grew it in cure culture, probably because the bug produces its own bacteriocins that killed off the competition. Okay, so this leads us to the big question. How do you treat it? And this is where things get complicated, because there's a real lack of specific guidance from major groups, whether you're looking at the Dutch guidelines from SWAB or international ones from bodies like EUCAST. So what are we supposed to do? Well, digging through the literature, a general consensus emerges. For isolates that are susceptible, high-dose beta-lactams are the way to go. For something like endocarditis, you're looking at a long course, 4 to 6 weeks, of high-dose ampicillin or penicillin. For abdominal infections, something with broader coverage like piperacillin tazobactam makes sense. But, and this is crucial, nothing is more important than source control. You have to drain that abscess. Now with kids, it's a real paradox. They're often given these probiotics, especially preemies in the ICU, but they are also the most vulnerable to getting a serious infection from them. The treatment approach is pretty similar, weight-based ampicillin, usually, but the key here is to have a high index of suspicion. If you have a septic child who's on probiotics and they're not getting better on empiric vancomycin, you've got to be thinking about this bug. So let me be perfectly clear on this. The bottom line is, you can't guess. You cannot assume it's going to be susceptible to penicillin. Resistance has been seen. That's why running antimicrobial susceptibility testing, or AST, on every single clinically significant isolate is absolutely non-negotiable. And right here is where we hit the biggest wall as a clinical lab. Okay, so we run the AST, we get an MIC value, a number. But what do we do with that number? What does it actually mean for the patient?
This is the critical gap. There's a delay between when we start recognizing a bug as a real pathogen and when we get the standardized tools to deal with it. Without official clinical breakpoints from an organization like EUCAST, we can't confidently label an isolate as susceptible, intermediate, or resistant.
So what do you do in practice? Your reporting has to be very careful and interpretive. First, you have to state clearly on the report, no clinical breakpoints are available. Then you give the clinician the raw MIC value. The best thing to do after that is compare the MIC to the epidemiological cutoff, or ECOF. That lets you tell them if the bug is wild type or if it might have acquired resistance. And finally, you have to add a comment recommending they correlate clinically and talk to an infectious disease expert.
So that brings us to the last and maybe most important part of the puzzle, prevention. How do we stop these infections from happening in the first place, especially when, a lot of the time, we're the ones introducing the risk?
The biggest issue here is what we call iatrogenic risk. That's harm caused by medical treatment. Think about it. We are giving live, intrinsically vancomycin-resistant bacteria to our sickest patients. For anyone with a compromised gut, after surgery or with mucositis from chemo, the risk of that bug moving into the bloodstream is very, very real. Not to mention, cracking open those capsules at the bedside can contaminate everything, including central line hubs. The takeaway is simple. Hospitals need policies that strongly discourage, if not outright ban, the use of probiotics in the ICU and for immunocompromised patients. And this leaves us with a huge challenge for the future, one that goes right to the heart of patient safety. As the use of probiotics in medicine keeps growing, how are we, in the lab, in infection control, in pharmacy, going to adapt to this threat that we're creating ourselves?
Everything we talked about today was pulled together from a Gemini deep search. If you really want to get into the weeds on this, I highly recommend checking out the full notebook document. You can find all the references and source materials linked there.
But now, I really want to hear from you. Have you seen Ligilactobacillus salivarius in your lab? What kind of policies does your hospital have around probiotics? Let us know your thoughts and experiences down in the comments. And of course, for more deep dives like this one, make sure you subscribe to the channel. Thanks for watching.